Welcome to Syngene DMPK Labs, where innovation meets precision. Here, we harness the latest technology to deliver timely and a high-quality data that fuels the journey of a novel chemical and biological entity across in silico, in vitro, and in vivo stages, as well as modeling capabilities. Our goal is to empower you with the insights needed to make a well-informed decisions at every milestone of drug discovery and early development. At Syngene, DMPK isn't just a process. It's the dot well connected across hit identification, lead optimization, and candidate selection. We focus on translatable pharmacokinetics for an actionable insights, supporting your quick to clinic approach. Our commitment to excellence drives us to invest in end-to-end automation across our central compound management system that can host 400,000 compounds to fully robotic high throughput sample preparation using 96 and 384 well plate formats. We strive for efficiency. A fleet of advanced mass spectrometers, including triple quadruple API 7500, EcoTalk, OrbitTrap, and QTrap paired with UPLCs ensures that we deliver nothing but the best. Plus, our automated data analysis and reporting tools are tailored to meet Syngene and our client-specific formats. We ensure full traceability and an easy retrieval of all the work through electronic laboratory networks and audit trail-enabled data acquisition systems. And with our unique DIY e-commerce platform, SignMap, where you can choose an assay, order online, and track, ensures collaborating with us has never been easier. Let's work together for troubleshooting druggability issues and pave the way for a successful drug discovery.
We at Syngene have a state-of-the-art GLP-certified and ALAC-accredited vivarium, which is one of the largest vivarium in India.
Animals used for experiments are procured from international breeders and are housed in IVC cages using the highest animal welfare standards. A team of brilliant, highly motivated, and skilled scientists perform all survival surgeries in-house, and we conduct more than 4,000 PK studies every year with hands-on expertise in pre-formulation development. Our team has successfully predicted human PK parameters of NCEs and predicted first-in-human dose, which helped our clients to progress their molecules in clinical trials at faster rate.
It's truly a privilege to lead a team of incredibly talented and passionate scientists who tackle bioanalytical challenges for our clients across the globe, day in and day out.
Our labs are equipped with cutting-edge infrastructure, from advanced liquid handling systems for automated sample processing to state-of-the-art LC-MS instruments like the API 7500. These tools allow us to push beyond conventional detection limits, reaching nano, pico, and even femtogram levels, not just in plasma, but across more than 20 different biological tissues. We're here to support all your bioanalytical needs, from serial sampling to PROTACs, with deep expertise in chiral analysis, LC-MS assays, and advanced biomarker techniques.
We have the advanced technologies to handle the wave 1 in vitro ADME assays in fully automation mode, and we ran more than 40,000 assays in a year.
At every stage of drug discovery and development, we conduct comprehensive assessments of druggable properties, such as solubility, lipophilicity, permeability, both artificial and cellular metabolic clearance, enzyme interactions, CYP profiles, biotransformation pathways, and structural soft spots. Our work is deeply collaborative, integrating closely with chemistry, biology, and toxicology teams. Leveraging advanced technologies, including Q-Trap, TOF, and OrbitTrap, we support research on small molecules, PROTACs, peptides, peptide drug conjugates, molecular glues, and antibody drug conjugates.
Okay, so welcome to Syngene's scientific webinar series. This is your moderator, Vishwottam Kandikere. Welcome to all our audiences, and it's indeed a great privilege to see you all in our sequential webinar series. We have a few interesting scientific thought processes which we want to share with you. We have few eminent speakers who are going to talk on some of the key and hot topics today. Indeed, it's our privilege to bring such a valuable seminar which builds the scientific connectivity and also the scientific trust across creating something novel and developing compounds which is close to building medicines for the patients in need. Let's go over today's agenda. Hope we're able to see the slide deck. Indeed, the scientific webinar series named DMPK Unravelled has been our ambitious proposal to bring the academicians and the global researchers and our collaborators on a same platform to exchange some of the advances in life of discovery. Okay, now as the time clicks and ticks, we would like to brief upon the vision of DMPK Unravelled, and there are certain webinar etiquettes. All of you have joined as an attendee mode. If you have any questions, we request you to please post on your chat box. We would like to discuss them, and we have allocated a time at the end of the webinar where our eminent speakers will discuss and also will be able to share their perspectives on that. We have two eminent speakers today. The first speaker is Harish Kaushik Kotakonda, who will be talking on solving ADMET challenges in antibiotic development, and Rambabu Patem, who will be talking more on a role of DMPK in a prodrug development. Next slide. Well, let me take a couple of minutes briefly on speaking about Syngene. So Syngene is one of the largest contract research organization and is a partner of choice for global clients, providing contract research, development, and manufacturing solutions. Syngene is set up over more than 2 million square feet of an infrastructure and hosts high caliber scientists of more than 8,500 as a global workforce, has been on a stringent triangle of quality, safety, integrity, encompassing and passing through more than 200 client audits and also endless regulatory audits. Syngene focuses on ESG part of it, making sure more than 75% of power is from green. And more than that, Syngene is contributing to the drug discovery and development over the last 30 years. Next slide. Well, Syngene has been working on various therapeutic segments and modalities across small molecules, oligonucleotides, antibodies, antibody drug conjugates, proteolytic targeting chimeras, and CAR T therapies. There has been multiple therapeutic areas. It has been spread its wings. A few of them are listed there. However, it has been a great privilege working with more than 450 clientele across the globe on these modalities and therapeutic areas. We would be excited to share some of those case studies in these webinar series in upcoming sessions. Well, the DMPK Unravelled webinar series is an exciting platform designed to bridge the gap between the academia and industry by sharing valuable insights and success stories in the field of DMPK. The series aims to bring together thought leaders, academicians, industry scientists, and researchers across the globe to discuss the latest advancements in DMPK, with a focus of overcoming key challenges such as drugability issues and optimizing the molecules for the next stage of drug development. They highlight real-world case studies, practical solutions. The series will foster collaboration and knowledge exchange, empowering participants to navigate the complexities of drug discovery and development more efficiently. Well, we have been discussing the challenges of drugability over the last 30 years. When we look at these graphs, the DMPK has been understanding the drugability challenges, discovery challenges, moving compounds from compounds from a chemical to a medicine through a long marathon ride with a lot of hurdles of drugability. The global DMPK scientific fraternity have come through devising strategies of right molecule at the right time at the right assay to ensure the drugability challenges are optimized. So there has been an apparent improvement in the DMPK attributed attrition, but still there is a significant, can be attributed to various aspects. It can be efficacy, can be formulation, cost of goods, toxicological reasons. But remember, those who do not know history are destined to repeat it. So we have case studies, we discuss the scientific thought process to ensure we engage the right assays at the right time to devise the right mitigation plan. There has been various strategies and there has been various adaptive flow schemes. How do we move from a hit to all the way up to candidate stage? There are various in vitro ADME assays. There are various in vivo assays across the allometric species. The DMPK scientists co-work with efficacy pharmacology, formulation development, toxicologists, and iterative SAR cycles according to the DMTA planning. DMPK has been defining itself by the knowledge derivation while working with all of them and adding a high value design making to a pipeline. Now, unraveling the DMPK secrets has also been our objective. Setting this as our core objective, let me introduce our first speaker. Harish Kaushik Kotakonda. He's the Director of DMPK and Clinical Pharmacology at Bugworks Research Incorporated, where he heads the DMPK function and drives drug discovery of novel antibiotics and anti-cancer compounds. Bugworks has been instrumental over the many years now, instituted by passionate drug discovery scientists. And Kaushik has been instrumental in driving many of the programs to the clinical stage. And I assume it's more than 5 compounds which has come to the clinical stage driven by the Kaushik. He has published an innumerable number of publications, guided many research graduates for their PhD and MS level scientists. And he has a wealth of experience in driving drug discovery. I welcome Harish to present his one of the coveted talks on solving ADMET challenges in antibiotic development, the real-world DMPK strategies and translational successes. Welcome, Harish.
Thanks, Rishi. Thanks for the kind introduction. It's a pleasure to present my perspectives and thoughts on how DMPK enables drug discovery. And we are going to discuss further on this particular aspect. Next slide. So, standard disclaimer slide. Next slide. So this is going to be my outline of my presentation. We want to talk about why antibiotic innovation is actually urgently needed. And in this process, what at Bugworks, what we have done to discover BWC 977, a broad spectrum novel class, IV and oral route of administration possibility. And in this process, how we overcome different developmental barriers, both in terms of DMPK and also in terms of PKPD. And what are the different the model-based strategies we have adopted in order to identify the right formulation, let it be, or understanding the translation aspects on the molecule. So we're going to talk about each one of these in my couple of coming slides. Next slide. So antibiotics, as we all know, without antibiotics, there is nothing with respect to the surgical success, which the surgeons greatly boast about, because a surgeon can do a surgery, but if the antibiotics doesn't do their job, unfortunately, the patient is definitely going to contract some infections, and then this is going to create a very serious issue. And so that's exactly the reason why antibiotics are considered to be the cornerstone of modern medicine. All these treatment modalities, what we have been looking at, will not be possible if antibiotics don't function properly. So that's the essence of antibiotics. And antibiotics has been one of the neglected areas just because of the beautiful translation. The science has been evolving over a period of time, but unfortunately, antibiotics need some special attention right now because we are amidst a very global pandemic. So what is causing this particular antibiotics failure in the clinical setup, or let's say in the hospitals, ICU hospitals, in a neonatal patient population? What is causing that? So when we look at the science behind it, it's actually the gram-negative pathogens, which are called as superbugs. There are 5 superbugs which are so-called called as escape pathogens. So all over the world, the US CDC and WHO has classified some of these pathogens into a different category called as critical, high, and priority pathogens. So all these gram-negative pathogens and some of the gram-positive pathogens as well have become resistant to the existing antibiotics, and due to which what's happening is that we are seeing deaths happening in the critical care setups, particularly in the ICUs. The patients are succumbing to the infections, let it be a bloodstream infections or let it be a lung-related infections. Okay, none of them are safe because of these infections, and all these is because of these 5 superbugs becoming resistant to the antibiotics which were discovered some time ago, and also which are being discovered right now and a couple of years ago as well. So that's the problem of antimicrobial resistance, which is a growing concern, and all the governments have laid an emphasis on putting some efforts in order to overcome this AMR crisis. And as at Bugworks, we also recognize this as one of the serious problem, and we have been working on this particular aspect from 2014 onwards. We have been working on this particular aspect. Next slide. Next slide. So where we are now, when we just walk back in the history, we understand that the first series of broad spectrum novel antibiotics were discovered in somewhere about 1960s. Fluoroquinolones were the first class of broad spectrum antibiotics which were discovered. Prior to that, there were beta-lactamase inhibitors. There were different kind of class of molecules. Cephalosporins were all there, but all these molecules were discovered some time ago. It is not that in the last, let's say, 40, 50 years, we have not seen any new broad spectrum antibiotic with a novel class have been discovered, and whatever have been discovered have all become resistant to the existing. The pathogens have become resistant to these antibiotics. So what's the issue when you look at when you actually dig into this particular aspect, and when you try to understand, it has got multiple reasons why we have not have a healthy R&D pipeline of molecules coming out of different organizations or different academic institutes to bring out some good antibiotics. There are a lot of issues in this, but this is a serious concern that we don't have a good broad spectrum class still discovered yet. So at Bugworks, as I've mentioned, we have been working on this particular aspect of solving this AMR crisis, global AMR crisis. And in this process, we have been working on discovering novel class of antibiotics it could be a broad spectrum antibiotics. So our emphasis right from day one was, let's work on identifying some broad spectrum molecules, broad spectrum antibiotics, which is going to help us in overcoming this AMR crisis. In this pursuit of discovery, what we have identified is a couple of molecules. One such molecule is BWC0977, which we are going to talk about today, which is an IV, which is administered through an IV route of administration, and is a novel class with a broad spectrum profile. And the same molecule, there is also a potential, because of its good oral bioavailability, that it can be also be administered through an oral route of administration. And there are a lot of other strategies which we think that it could help us to bring out the same molecule for different type of indications. So that's about our discovery pipeline currently. And then we have a very healthy pipeline of antibiotics coming through. So in the process of discovery of 977, we have been working on handling a lot of important aspects with respect to the safety part and also the microbiology potency. So this is all about the microbiological potency, which we are showing here. Let me just bring up my pointer. Yeah. So these are all the microbiological spectrum against the five different pathogens which I've mentioned about escape pathogens. And we also consider, because this class of compounds has got indication of a hERG potential. So we always track the hERG potential when we started this synthesis of these molecules using medicinal chemistry. So the emphasis was on the mitigating the hERG aspect, and also balancing the spectrum, particularly with respect to these five escape pathogens. And in addition to that, very important, as Vishu has mentioned in some slides ago, that the important ADMET aspects are critical to understand whether whatever these molecules we are seeing on the screen, can they be converted into a drug. So in this particular process, the different ADMET parameters have been optimized, and then that's how we identified that BWC977 ticks all the boxes in terms of both spectrum and the safety, and also the DMPK aspects. So when you look at 977, this is about the microbiological species testing, which was done in global setting. Something about 3,000 to 4,000 strains have been tested against 977 across the globe. Different strains have been tested against 977. We could see that 977 displays a very good broad spectrum activity, and not only by us, this molecule was also tested against in the microbiological species by different collaborators, because we work with international collaborators, like for example WHO and CARBEX. So they took this molecule and they tested in their own hands with respect to understanding whether this particular spectrum, what we have identified, is it true, and in their own hands they've tested it, and what came out is that definitely this 977 molecule is a broad spectrum against a very important priority pathogen list, which was shared by World Health Organization in 2024. All the five escape pathogens, what we could see on here. So these are all the four different pathogens which are very critical, and they are responsible, gram-negative pathogens responsible to drive the AMR crisis across the globe. And so to start with, when we started to understand 977, as you have seen 2 slides ago, that different ADMET aspects also we try to understand in this particular process. Now, one of the important aspects is to understand the metabolism part, and all these experiments were done using Syngene as a primary collaborator. So a lot of these experiments on understanding the human metabolism, and also the rat liver metabolism in human and dog liver fractions. So we try to understand what is the metabolism aspects of this particular molecule. In this process, we understood that there are two key metabolites because of the oxidation and degradation mechanism. And then we wanted to understand whether this particular metabolites, what we see in the preclinical species, do they really translate into the humans? So that was the objective, whether behind actually understanding the metabolism aspects of 977. And in addition to these two metabolites, 1903 and 1116, we also understood that oxidative ND-alkylation also is possible for this particular molecule. And then we expected that 1724 is another metabolite which may come across as one of the possible putative metabolites. And all these experiments have been done both in the combination of in vitro systems. So when we look at the linear IV pharmacokinetics, this particular molecule has been actually characterized across preclinical species, different species were used to understand the IV and oral pharmacokinetics. So this slide talks about the IV kinetics of this molecule, well-behaved molecule across the four different species. What we have taken up to understand the pharmacokinetics of this molecule in a preclinical species, following an IV route of administration. And this becomes a very important data set for us that this data actually has been used for us to perform a translation into the humans and do a modeling and simulation and prediction of the human dose from this particular data. So this becomes a very key data set which we have generated using Syngene and other collaborator. So this was one of the slides which I was trying to explain you that the metabolism aspect, what we understood in the preclinical in vitro systems. So we wanted to see whether the same thing is observed in both in the in vivo system, particularly in the preclinical in vivo system. So this particular data set, it's about the bile duct cannulated animals, which this experiment was done at Syngene. So we perform bile duct cannulation, and then this is we administer the compound, and we try to quantify post IV route of administration in different matrix. It could be a bile, it could be plasma, it could be urine, it could be feces. Different matrix was collected, and then we try to understand whether the metabolism aspects, whatever we saw, whether they actually contribute to the significant percentage exposure compared to the parent. So this is the data what we have, and we found that 977 is actually significantly present compared to the metabolites. All these metabolites contribute to less than 5% of the exposure of 977, which makes that 977 is only the key compound, and the metabolites doesn't contribute too much. So what also it also says is that what we hypothesize that metabolism could be the hepatic metabolism could be one of the key factor in terms of metabolism. We did not observe that metabolites happening in the in vivo system, particularly in the rat. And similar thing, what we did in the humans as well, as part of our first in human study, we did some exploratory analysis. This is not a hard, it's not a standard protocol of using a cold compound using an ADME studies, but this is an exploratory measure. We collected the plasma and the urine of the volunteers participating in the trial at the high dose, and then we try to understand whether these metabolites, what we think are putative metabolites, whether do they really form in the humans. So what we understood was that whatever we observed in the rat, the same phenomena has been observed in the humans. 977 being the primary compound analyte observed in the different matrices compared to the metabolites. So it clearly tells us that the molecule is actually translating well between the species, and also that we also understood that metabolism is not the key aspect here. There is a different route of elimination. Elimination could be happening from the different route. It could be urinary route of excretion. So that could be one of the major route of elimination of this particular molecule happening from the in vivo system, but it's not metabolism aspect. So the other important aspect is that to understand the PK/PD index of an antibiotic drug. So just for the benefit of all the people who just wanted to understand the PK/PD aspects of antibiotic drug, there are three key PK/PD indices which we generally talk about in the antibiotic drug discovery world. One is called as Cmax over MIC. It's basically the MIC is the minimum inhibitory concentration, which we measure using the microbiological MIC testing happening in the test tubes and the cultures. So we evaluate the MIC of our drug across the different species, and that's the benchmark. So we wanted to maintain the PK of our molecule in terms of these three parameters called Cmax over MIC. Cmax, what is the peak maximum concentration which is above the MIC, and then also the exposure over MIC, which is AUC over MIC. This is also one of the key PK/PD index for some of molecules, and then also time over MIC. So these are the three key PK/PD indices which generally we try to understand when we actually start work on antibiotic drug discovery, and we try to correlate the PD with the PK and try to understand, tease out the key PK/PD index, and then basing on the PK/PD index, we try to establish the doses going forward downstream. So it's very important for us to understand that in the antibiotic world, PK plays a very, very important role in terms of correlating the PD aspects, and so that's a very important information we try to derive in the preclinical studies. So this is the slide which talks about what we have done is that over a period of studies, we determined that AUC over MIC was a key PK/PD index for this molecule, and we did a lot of studies in terms of animal models, and then we derived that we could actually understand that for a combination of different pathogens, we derived the AUC over MIC. What is the required AUC over MIC which is needed to for this molecule to demonstrate the efficacy? And all this work has been published in Nature Communications. We also understood that this molecule is highly concentrated when we administer it in the lung compartment, which makes this molecule demonstrate its exposures readily available in the required target site of infection. Let it be a pneumonia indication where lung is a critical site of infection, and having this molecule demonstrate dedicated target distribution in terms of lungs, which is also very helpful for us to highlight that it could be a pneumonia indication drug. So these are all about the animal models, what we have done over a period of time: mice thigh infection model, rat thigh infection model, and rat lung infection model. So we collated different pathogens and then determined the PK/PD index here. So we could see that AUC or MIC, these are the ranges what we think is needed to maintain the efficacy of this particular molecule. Then comes the what, as I've told you, the preclinical data is absolutely gold for us, because that actually really helped us to understand the translation. And this has been there in the DMPK world in terms of allometric scaling, which people have been following it. What we tried to do differently is that because we had different species data, we fit this entire data into a compartment model, like a PK compartment model. In our case, it was 2-compartment model. So we fit the data to a 2-compartment model. And then we used a population modeling approach where we try to include the entire all the data sets of individual species as well in terms of building a population model. And then we use the basics of allometry coupled with the population model, and we try to understand the human predicted equivalent dose. So we use that particular algorithm to understand what will be the human dose in terms of when we go into the first in human study, what should the dose I should target? If this is the dose, what is the Cmax? If, let's say, what is the top dose? What is the mid dose? What is the highest dose I should go, so that I can maintain the Cmax and I can achieve the required AUCs. So then also we also did a different way of approach of the very popular approach of using physiologically based PK modeling approach. So the entire data, preclinical data, was used to build a PBPK model, and then that particular model was translated into the humans. So there are two different approaches we used. One is the population-based, model-based allometric scaling approach, and the other one is a PBPK approach. And then, to our pleasant surprise, we were very happy to see that when the clinical data started coming to us, we could see that whatever we have predicted in terms of the translation aspect, it really came true. So this you can see here, the below two slide talks about the observed Cmax versus predicted Cmax and the R-square values. So it actually shows that how beautifully this molecule is translating from the animals to the humans, and also the better application of a right approach of modeling approach will help us to understand a lot of aspects in terms of translation. So this is a human phase 1 data. We just completed our phase 1 data, first in human single ascending dose study. So we completed a single ascending dose starting from 120 mg to 1050 mg. And what we observed from the clinical data was that this compound exhibits a moderate systemic clearance across the single dose levels. And it's highly distributed with a high volume of distribution with a good T-half. And overall, when we look at the dose linearity, we could see that there's a good linearity happening across the 5 dose levels in the single ascending dose. Multiple ascending dose, we are actually we are yet to start that. We're going to start that multiple ascending dose trials very shortly. And then, so what we did was that, okay, now we got the human PK data from the single ascending dose. What could be done with this particular data was that we wanted to use this particular data, the human PK data, first to set up the required multiple ascending doses. We wanted to understand what is the targeted dose, because we have only three or four cohorts. So we wanted to use those 3 or 4 cohorts judiciously, wisely, in terms of identifying the right dose, which will give us maximum information in terms of taking that particular dose levels to the phase 2s and phase 3s. So what we did was that we tried to fit the data to a population model, and this is the population model algorithm, and the estimates what we have got. So using the population model data, what we have done is that we have established the required doses to go into the multiple ascending dose cohorts, the 3-4 doses, which is going to translate into the efficacious doses. So we have identified those doses, and this particular model will be further refined once we get the model multiple ascending dose. And this is going to be the bedrock for us in terms of modeling and simulation, because this is going to be very useful in order to perform a PK/PD analysis on the human PK data and the human PD data, also to understand the probability of target attainment. This is one of the key metrics which we try to do in antibiotic discovery world. So coming to the oral therapy, so this molecule has got some of the unique DMPK challenges, because of which, as we all know, nowadays we don't get to work with any BCS class 1 kind of a compounds. Whatever we have, it's all BCS class 2 kind of or BCS class 4 kind of a molecules, because of the intricate chemistry which is involved, and chemists are trying to bring some IP innovations in designing the chemical compounds. So as part of that, 977 also has got some of the unique challenges, which as a DMPK scientist, we need to overcome. One is that low solubility. Because of its low solubility and then low permeability, it is classified under BCS class 4 compound. And it also has got a unique issue, because it's a weak base compound. It has got a pH dependent solubility and degradation. And it has also got a high Flex with a Flex ratio of 51 via PGP. Is what we think right now, but we are actually characterizing whether PGP is actually contributing to the effects, or are there any other transporters which are contributing to the effects. And then also CYP3A4 metabolism, the first pass effect. So we also did our DDI study, and we understood that clearly that following an oral route of administration, CYP3A4 comes into picture, and it is definitely going to hamper our oral exposures. And and because of its pH-dependent solubility, there is obviously a high chance that when the molecule undergoes transition in the GI environment, because of the pH changes, there is high probability that it's going to undergo precipitation. So a lot of aspects are there in terms of DMPK challenges, which we try to overcome using our modeling and simulation approach. And using the PBPK approach, what we did was that we tried to understand what are the key metrics which are important for us to understand and optimize further. And basing on that, we try to design the formulation in such a way that the formulation will help us to overcome all these challenges. In this process, we identified a sets-based formulation could be one of the good approach in order to overcome these DMPK challenges, and also to improve the oral absorption across the GI tract conditions. And this is about the oral efficacy part. So when you can see that through an oral route of administration also, this molecule demonstrates similar efficacy as what we have seen in the IV. And it's actually very rare to find a molecule with a broad spectrum molecule with an IV route of administration, and as a step down, having an oral route of administration. It's very, very rare these days. You can actually look back into the literature in past 40-50 years. You don't have any molecule which is an IV and having a step down oral option. Either it could be an IV molecule or it could be an oral molecule. But this is the first time we are having a molecule which is having an IV potential followed by a oral step down potential. So that is very innovative aspects of this particular molecule. And if you look at the WHO clinical pipeline analysis, as I told you, there is no such molecules which are actually which is ticking all the boxes in terms of broad spectrum. And 977 sits right on the top of those top 5% of novel broad spectrum antibiotics in terms of having an IV and also an oral step down option. There are very, very, very few molecules available in the clinical pipeline as determined by World Health Organization in terms of what is there coming in terms of the clinical pipeline R&D stages, which can translate into the phase 2s and phase 3s and then ultimately a drug. So in those aspects, 977 is one of the mostly highly regarded, mostly highly awaited molecule in terms of antibiotic spectrum, and also having a two different potentials of IV and oral step down option. So what we have done over a period of 6-7 years is that we try to identify a novel broad spectrum molecule, and in this process we try to overcome one by one the DMPK barriers we faced in terms of oral delivery of a broad spectrum agent. As we have seen, there are a lot of challenges in terms of bringing an oral absorption happening of this molecule. So we try to understand that and we try to overcome that. And we have been using very consistently the modeling-based approach right from the initial preclinical stages right to the clinical stages as well. Modeling has been always a guiding light in terms of taking the translation aspect. And as I told you, it addresses, this molecule addresses the critical unmet need of resistant infections with IV to oral step-down potential. And we are going to, once we complete a multiple ascending dose, we have identified that this molecule can be studied further in a phase 2 trial for a serious bacterial infection indication. So this is a pivotal role of PKPD translation. As I've told you, PKPD makes a very, very important toolbox in terms of antibacterial development. There is no hidden fact that without PKPD understanding, there is none of the molecules are being discovered in terms of antibiotic development. So this is the slide which talks about what are the key PKPD data which is used for. So I'm going to explain that in terms of a case study of a molecule which is just recently discovered and approved. So this particular molecule called cefepime enmetazobactam, this has been discovered by Orchid Pharma from Chennai. They discovered this molecule and they took this molecule to certain stages of development. And after that, they actually outlicensed this molecule to Electra Therapeutics in Germany, and they completed their phase 2s and a phase 3 clinical trial. And then even Electra Therapeutics could not sustain the financial burden of doing a clinical trial because of the anti-infectives, no revenue potential, no investment as well. So they had to hand it over to a different company, different organization, and they took this approval, NDA approval from global regulatory agencies. And now this particular molecule has been approved. And so if you look at this particular molecule, what these guys have intelligently did is that they did a nosocomial pneumonia indication. They did try to understand the exposure of this molecule in the lung tissue, particularly epithelial lining fluid of the patient population. And they understood that in the lung compartment, the exposure of the drug is huge compared to the plasma. That gave them a little edge, demonstrating that in the required tissue compartment, site of infection compartment, the exposures are very high. So using this particular data, what they did is that they again used a PKPD modeling and simulation. They used the clinical PK data and also the PD data from the preclinical species, and they try to use the PKPD approach. And then they identified that if this particular molecule is administered at this particular dose over a period of 2 hours time frame, then this particular molecule, cefepime enmetazobactam, can also be used in terms of pneumonia indication. So what they have done further is that when you look at the US FDA label, so in the US FDA, the initial phase 3 trial was done only in the CUTI versus piptazo. This is another standard of care in terms of phase 3 trial. They did this particular trial, and then US FDA goes by indication. It doesn't go by, let's say, target pathogen infection. It goes by indication. For this particular indication, if you want to get a label approval, you do a phase 3 trial. If you want to go, if you want a pneumonia indication, then you do a phase 3 trial. That's how the US FDA thinks. So what they have done is that they have did a CUTI, a complicated urinary tract infection trial, a phase 3 trial in 1,041 CUTI patients versus piptazo, and they demonstrated that clearly that this molecule has got a good benefit in terms of overcoming the CUTI infection, and that's how US FDA also has given a label indication of a CUTI for them. Now, in contrast, when you look at the EMA, just because one particular trial it's not going back. Yeah. So as I've just discussed in the previous slide, because they did one nosocomial pneumonia lung exposure trial. So with that particular data, coupled with the CUTI data, what they did is they took these two data packages to EMA, and EMA considers any antibiotic data package slightly differently compared to US FDA. So EMA, what they have considered is that because this particular molecule targets against those critical pathogens, and it is also accumulated well in the required lung compartment. So what they have done is that they obviously gave a CUTI approval because the phase 3 trial was in CUTI, and they demonstrated a margin of success in terms of phase 3 trial. So obviously they got a CUTI label indication. In addition to that, they also got a HAP-VAP, hospital-acquired bacterial pneumonia, and ventilator-associated bacterial pneumonia indication as well, just basing on the modeling and simulation of the data, or the phase 1 data in 19 healthy volunteers. They did that. as I've talked about in the previous slide, where they demonstrated that the exposure of the molecule is very good in the lungs. So using that particular data and coupled with the safety data coming out from the phase 3 trial, so EMA has given approval of label indication approval for HAP-VAP. In addition to that, serious bloodstream infection as well. So EMA gave them a broader label approval compared to US FDA. And also recently DCA also has given approval to this molecule. So shortly this molecule will be in the market. Thanks to Orchid. Orchid is in-licensing this molecule back into their hands. And then they are actually bringing this molecule into the market. So that's about this molecule. So you could understand how PKPD is actually critical at every stage of development, be it the preclinical stage, or let it be the translation stage, or in fact in the actual critical pivotal phase 3 stage as well. How critical PKPD modeling and simulation, understanding on a lot of DMPK aspects are very, very important for us to actually bring a molecule, a good antibiotic molecule to the market. So just to summarize, finally, our clinical candidate is called as Gyrox class of compounds, a novel class of antibiotic with 977 as a front runner. It's equipotent against two important targets called topoisomerase 4 and DNA gyrase. And we tried to raise a resistance to this particular molecule. We tried to raise it. Unfortunately or fortunately, we don't have any resistance. Despite passaging to a 4x MICs up to a month, did not yield any target site mutants. And if you look at the indications, what we have targeted is, it acts against the hospital-acquired infections in terms of different pathogens, biothreat as well, because biothreat is also one of the key worry area for US government in terms of chemical warfare. Bacillus anthracis, as everybody knows, is one of the pathogens which is used very commonly in terms of chemical warfare. And so this molecule is also active against those biothreat pathogens. In addition to that, cystic fibrosis causing pathogens as well. So it's basically one molecule is going to solve a lot of infection conditions. So as I've mentioned, we completed a phase 1 trial with a linear PK and adequate urine exposure. So in the urine compartment, a good amount of drug is being observed. Unchanged drug is getting collected in the urine, with about 30% of the drug is getting excreted through the urine. And as I've told you, we need to do an ADMET trial to understand the different elimination mechanisms further. So we believe that this is going to be the first novel broad-spectrum antibiotic in 6 decades, which is going to active against all MDR pathogens, one product label, multiple indications for critical care, community, biothreat, and with an oral step down to reduce the inpatient time at hospital. And this becomes a very critical aspect because in the Western world, the insurance companies rule the roost there. So they don't want patients to stay in the hospital for a longer time. They want to actually push out the patients from the hospital. Once they feel better, they want the patient to go out of the hospital. And it's also good for the patients as well. So having an oral step-down option actually reduces the hospitalization time. And it's also active against significantly harder to generate drug-resistant bacteria. So that's about our molecule. We believe that's going to be a breakthrough product addressing the needs of patients all over the world. And this is the team working at Bugworks who have been responsible over a period of time to bring out this wonderful molecule. Hopefully that this molecule will continue its successful translation across the phase 2s and phase 3s. So I think that's my last slide. Thanks.
Okay, so that's a fantastic journey, Harish. It's really phenomenal to see a world-class antibiotic coming post 6 decades. In fact, it has, my first drug discovery learning was antibiotics. I'm very glad to see after so long time, was a phenomenal, passionate outcome of a product. We would be more interested in hearing from you and understanding, and there are our audience has some very interesting questions. However, maybe we can club our question and answer session at the end of it. And let me thank you for a short while, and we will get back at the Q&A sessions. Let me take this opportunity to welcome our next speaker, Rambabu Patem. He is the Assistant Director and a Senior DMPK Leader at Syngene International. Rambabu has been working over drug discovery since last 23 years across small molecules, galanicals, biologics like monoclonal antibodies and ADCs. He has worked in various research organizations across India, and he's passionate about his research on biotransformation of small molecules, peptides, prodrugs, and advanced mass spectrometry studies across the paraphernalia of small molecule hit to IND enabling preclinical studies. He has also published extensively. Let me welcome Mr. Rambabu to present his talk.
Yeah.
Today, Rambabu will be presenting on aspects of prodrugs.
Good.
And over to Rambabu. Thank you.
Thank you, Dr. Vishwatam. Next slide. Yeah. So in this topic, I want to discuss majorly about the introduction to the DMPK part and even prodrugs and the objectives of the prodrug development. At the same time, the role of DMPK in the prodrug development, so which includes selection of the screening assays, development of the suitable analytical techniques, and what are the issues, how to handle it with one case study, and discussion on the further learnings as well as the improvements. So coming to the introduction to the DMPK, as we all know, DMPK is the drug metabolism and pharmacokinetics study. So basically, it will take care of the compound. So whenever there is a compound coming into the drug discovery or to market, usually people look at how it is giving the efficacy, how it is active, and how it will reduce my disease and everything. But nobody will cares about the what is the fate of the drug, but that will be taken care by the DMPK. So by studying various properties of the compound at different stages from starting from synthesis to the clinical level. So DMPK involves from the synthetic stage to the clinical level. That is usually bench to bed. So that involve number of steps. So which majorly involves the study of the physicochemical properties and stability of the compounds in various aqueous media like buffer, passive, passive, all those GI fluids. So and to ensure before going to the compound into the druggability stage, so to ensure all the properties are suitable for the druggable candidate. So at the same time in the DMPK we study all the about the absorption distribution studies which involve majorly with the protein binding and permeability studies. So those are all will be involved in the DMPK assessment. And at the same time, metabolism and DDI coming to the, so their in vitro systems will be used like microsomes and hepatocytes, S9 fractions. So these will be used to understand what is the fate of metabolism and how the metabolites will be formed. So those metabolites will be whether efficacious or it will be toxic or it is majorly for the elimination pathway to ease out the elimination of the drug from the body. Since it's the major requirement is once the efficacy and everything is done, it has to be eliminated from the body. So to study all those things, obviously DMPK is the major route. So that involves study of all these things. So these are majorly in vitro studies. So these in vitro studies involves the metabolite identification and fingerprinting. So where this will give indication, so whether the compound is getting metabolized and we need to do what are all the changes or modifications to be done to improve the stability of the compound in the metabolism as well as the in vitro studies. So further it will be done with the rodent and non-rodent PK studies. So as we have seen the previous case study presented by Harish. So in the DMPK it involves all the qualitative and quantitative bioanalysis with respect to using LCMS instrumentation or HPLC or GCMS or SFC. So different analytical techniques will be used. So coming to the prodrugs, prodrugs are the modified chemical form of an active pharmaceutical ingredient. So which will be further entered into the systemic circulation and it will be stable until it converts to the active form because of the in vivo biological systems, either through metabolism or maybe hydrolysis. So the prodrug object of the prodrug development majorly to improve the solubility, chemical stability, and permeability, lipophilicity to improve the permeability as well. So that may be passive permeability. So, and at the same time, from the metabolism point of view, to improve the enzymatic stability and permeability by adding some of the amino acids as the promoieties. So that will improve the carrier mediated transport, and it will improve the further bioavailability of the compound. And if there are any CYP liabilities, so to mitigate that, so there will be promoieties will be added, and those promoieties will be cleaved into at the site of action, where it will release the active compound, and that will show the action. So at the same time, to reduce the toxicity as well. So this is by targeting the compound or active moiety release at the target site. So these are some of the case studies I have included, where the role of DMPK in prodrug screening. So to understand the solubility, so this is one of the compound which is a paclitaxel. So it has the low solubility and low bioavailability in mouse. And when they are adding, when we are adding this disulfide bond, disulfide bridge, then it is a disulfide prodrug, and which is having the solubility, aqueous solubility more, and it has the even increased bioavailability up to 5-fold. So that is what it is happening with the prodrug approach. So these are all can be screened in the DMPK itself. And this is one more example what I have mentioned previously. So adding the glycine as the pro-moiety to the midodrine drug. So that is even further cleaved into the desglycine midodrine by peptidases, and it is the active moiety which is producing the activity. So this will increase the permeability, and thereby it is increasing the percentage bioavailability to more than 2-fold. And this is the one of the prodrug screening where we have seen improved stability from the metabolism point of view and absorption, as well as the low toxicity. So doxorubicin known as a cancer compound, and it causes cytotoxicity through various mechanisms, and its prodrug of amino acid, which is around 9 amino acids, it is undergoing different metabolic pathways through MMPs, and it is giving the 3 amino acid doxorubicin moiety, and it is further undergoing to the single leucine doxorubicin through proteinases, and that will further undergo metabolism through proteases, and it will release the doxorubicin at the site of action, so that it will reduce the toxicity, and it even increases the 10-fold bioavailability. And there are some other compounds which is associated in with the improved chemical stability and in the aqueous media, as well as the bisulfide prodrugs. So this is increasing the chemical stability. And with respect to the enzymatic stability of the enzymatic stability, valcyclovir is a prodrug of acyclovir. So this is also to improve the stability in the J lumen with respect to the enzymes. So this is the screening assess ADME evaluation. So where, as we are discussing previously, so this prodrug, which is the chemistry optimization by adding the promiotics to improve either solubility or stability and even toxicity point of view. But from the ADME point of view, we have to ensure whether this is superior to the active moiety when it is screened in the ADME evaluation point of view. So we need to ensure the solubility and stability of the compound in the aqueous media. And if there is, we have seen some of the compounds having the low or moderate solubility, then obviously we have to go for the prodrug optimization further. And at the same time, if it is having meeting our criteria, then obviously we have to move to the next step to improve to check the solubility, to check the stability in various biological matrices like intestinal S9, microsomes or hepatocytes, and all those things. And at the same time, we can study the permeability to improve whether it is improved or not. So this is all based on the required target point of view from the ADME evaluation. So we need to understand the druggable property of the compound to be improved, and accordingly we have to select the studies. So at the same time, we have to study the plasma stability. So prodrugs are known to have instability in the ex vivo conditions. So in that cases, we need to optimize the prodrugs to stabilize in the plasma by adding some of the stabilizers like sodium fluoride and PMSF, all those things. So then further it can be taken to the in vivo PK evaluation followed by the clinical. So this stabilization has to be performed before going into the PK evaluation. So here I want to mainly touch upon the bioanalytical method challenges. So for the prodrug ADME evaluation, which is maybe similar to the small molecules like Protax. So but in case of bioanalytical methodology, we have to be very careful to understand what are the impacts or what are the factors to be considered during method development, as well as the running of the any samples from the studies. So the majorly we can see the ex vivo instability. So this can be avoided by, this may be in aqueous media as well as the plasma biological matrices. In the aqueous media, this can be avoided by using the different pH conditions and temperature control during the processing. So that is to avoid the enzymatic stability, and by addition of the some of the sodium fluoride and PMSF to prevent any hydrolysis. And at the same time, rapid processing. So in the biological samples or any in vitro ADME experiments or in PK studies, we need to process the samples to put it into the LCMS. That means we have to clean the samples to go before the LCMS analysis. So this process will take time. So to minimize those things, we may need to optimize the processing time by taking minimum batches and all those things, so that in that cases we can avoid ex vivo instability point of view. And from the second point of view, distinguishing the prodrug and metabolite. So in LCMS cases, so we have to use the tandem LCMS based, so which is the more specific towards any compound or any particular metabolite or prodrug. And even sometimes we can use the stable isotope labeled compounds to understand. In case of metabolite, if you use the stable isotope labeled compound, so that will give indication where the metabolite is coming. So in that way, we can at least understand what is happening, if there are any issues, if there are any peak measures, that can be understood. And from the large molecule point of view, there will be complex large molecules like ADCs and PDCs as well, that is peptide drug conjugates. So with metabolites, so those metabolites are nothing but the payloads, so which is included in the ADC point of view. So these ADCs can be assayed using the ligand binding assays or maybe LCMS or combination of these two. So in these studies, we can also use the immunocapturing followed by the LCMS analysis as well. And with respect to the other consideration, so ex vivo conversion, which we were discussing previously. So this is prior evaluation is important to understand and to inhibit any ex vivo conversion to have the positive data. And even incurred resample analysis. So this is most important to understand our method reliabilities and everything. And at the same time, regulatory expectations. So development and validation of the analytical methods to understand whether prodrug is causing some effect or maybe drug is causing some effect. So the method should be rugged to understand and to meet the regulatory requirements. This is one of the case study I have mentioned here with respect to the LCMS analysis, ion source fragmentation. So this is in-source fragmentation will happen in case of the LCMS analysis. So here the example is phenacetin, which is a prodrug of paracetamol. Even phenacetin is also active, but I have taken this example to discuss about what is causing in the in-source fragmentation. So this phenacetin will undergo metabolism and it will release the paracetamol, which is the active component. So in the chromatography, when we inject phenacetin, we can see the peak at 2 minutes, 1.99 minutes, and the paracetamol peak eluting at the 1.8 minutes. And there is one more peak at the RT similar to the phenacetin. So which is actually forming from the phenacetin due to the in-source fragmentation caused by the electronic voltages. So whenever we are putting any molecule into the LCMS, there will be ionization source where there will be electron voltages will be applied. So because of those electron voltages, this ethyl group is losing and it is producing the paracetamol same only. So since it is we are eluting on the column point of view, means LC column point of view. So in the same RT we can observe one more peak. So that is due to the ion in-source fragmentation. So these can be optimized using the minimal ionization source voltage. So, and even the compound related parameters like declustering potential. So when we optimize these things, obviously we can see whether minimal or maybe no in-source fragmentation. To conclude on these things, it is better to undergo chromatographic resolution to understand drug and prodrug evaluation and to report any false positive data. So these are the most important things we need to understand and we have to work on to improve the accuracy and reliability of our bioanalytical methodology as well as the bioanalytical data for any regulatory submission as well as the to take the drug or to take the compound to the next level. And this is the summary from the discussion as of now. Products are designed majorly to improve the bioavailability of the any active moiety which is having some of the drugability property problems, like maybe solubility and stability and even permeability as well. So in case of prodrugs, we can use the routine ADMET screening strategy with the caveat of understanding the instability of the prodrugs either in aqueous media or enzymatic media and to stabilize those prodrugs so before taking into the any experimentation. Thank you. So with that, I conclude from my topic.
That's a fantastic talk, Rambabu. In fact, we have encountered many challenges while optimizing the druggability of a prodrug and ensuring as one of the alternative chemistry approaches when we have a fantastic compound in hand, but DMPK or a druggability is a challenging aspect. So whether it is a solubility or whether it is a permeability driven or is it a chemical stability driven, this is fantastic. Let us move to the next section. That's Q&A. Thanks, Harish. You have been, answering the questions online, and I'm sure many of the questions the audience already has. But there are a few very interesting questions which maybe I would like your attention. So one of the question is, congratulations to PWC 0977. So it's showing a consistent PK across the species and has low inter-subject variability in the phase 1. Now, how much confidence does this gives you in dose predictions for phase 2 and beyond?
Yeah. So thanks, Vishu, for asking that question. So the most important aspect is that we observed linear PK, as you have rightly saw that in the humans, we saw that linear PK and across the species both in the preclinical, also in the humans as well. That's a linear PK we have observed with a very tight data. So what we are going, so we have already seen a good translation happening from preclinical to the humans, right? So with that confidence, what we also have is that if we use a population modeling approach where we understand the variability coming up from different sources. So if you understand the variability well, right upfront in the phase 1 stage itself, that will help us to understand the variability and then identify the right dose going forward for the phase 2, maybe using these approaches to actually estimate the recommended phase 2 dose selection, and followed by target attainment simulation as well in the complicated urinary tract infection population. So we are very confident that if you use right approaches, as we have mentioned, we can understand the variability and then we can actually have a good handle on the dose which is needed for the phase 2s and beyond trials.
So that's really interesting. So since you said also, the good modeling, I also want to draw your attention, like you have been speaking very passionately on PBPK and POPPK in guiding formulation and dosing decisions. Maybe if you want to share some of your learnings while developing this compound, PCS class 4 oral compounds.
Yeah, thanks Vishu for bringing out this. Very important aspect is that we actually had laid a very strong emphasis on understanding the PBPK aspect, because when we embarked on the oral formulation development, we were very sure that this molecule will not cut the oral route of administration because of its complex nature. It was in terms of DMPK properties. So what we did was that we actually used the IV PBPK model, what we developed during the IV development, and we incorporated the ADMET data, what we have obtained over a period of time, and then we use that particular data to understand the oral absorption. What are those key parameters which are going to influence the oral absorption in terms of parameter sensitivity analysis? We did right at the pre-formulation stage when we started developing the oral formulations work. So that's the stage where we understood that, okay, if we understood that the pH is going to be a very key factor for this particular molecule, because we all know that in the oral GI environment, there's going to be a pH gradation, and our molecule can't withstand beyond 4.5 pH. So it definitely creates a problem for us when we go beyond 4.5 pH, particularly in the intestine region, like duodenum to intestine region. We are going to definitely come across a pH range of more than 5. So that's the reason why we wanted to understand all these aspects virtually using an in silico PBPK based approach simulations. We did a lot of simulations, and that's how we understood that what are those parameters which are going to impact my Cmax, impact my AUC, and that's how we identified those parameters, and that's how we went behind those parameters to overcome each one of those factors. Like metabolism could be a key factor, so we wanted to understand how much impact the metabolism is going to make on the oral exposure. So we understood that it's going to be a very important factor. So then we brought in some of the excipient which can help us in order to overcome the metabolism aspect in terms of CYP3A4. So like this, we did a heavy reliance on PBPK modeling approach in order to particularly design oral formulation.
And that's really great learnings, Harish. Indeed, we have been seeing it's a fantastic collaboration with Bugworks and your team over so many years in bringing some of these decision-making experiments. It's really fantastic to listen to you on this talk. I have a couple of questions for Rambabu. Mr. Rambabu, you have been quite passionate and an expert on a prodrug. There are questions from some of our audiences. So if we are adding an amino acids to utilize transporters, so which in vitro tool is ideal for screening these products? And as co-expression of transporters is very much different than actual human intestine. What's your thought on that?
So thanks for the question. So basically, we can use some of the humanized or recombinant cells technology, where we can use the mate or some of the cell lines like BCP cell lines. So those can be used to understand how these will be how these will be working, and how these act to improve to understand the transporter related activities.
Oh, that's fine. That's good. Are there what would be the ways to improve permeability? Because permeability has been a very tough task, right? Prodrug, one of the challenges are either solubility or permeability. So in your learnings, in what ways we can improve the permeability?
So basically, with respect to the passive permeability, we have to increase the lipophilicity of the compound, so that will improve the passive permeability. But in case of if you want to go with any transporter mediator or maybe efflux transporters related some things, you have to majorly use the phosphate prodrugs, or maybe in combination with the amino acids. We have to try all those things. So that will improve the permeability of the prodrug compounds. And we have to understand before going into the experiment how this prodrug will be stable in that cell media, because there will be alkaline phosphatases which will break down the compound. So to understand those things, more important before going into those studies as well.
Fantastic. There has been an endless list of questions. Thanks to Harish, he has tried to answer online. I would like to update to all our audience. All the questions will be answered in more detail, and we would post that in Syngene's website with more elaborative answers in a couple of weeks. And also this recording live webinar will also be posted on the Syngene's website for a later view. This is very well available. We have already slightly overshot our time. We really appreciate all the science enthusiasts who have joined this call. I would like to extend my real thank you to all the audience who stayed with us for more than an hour now. Also, our eminent speakers, Dr. Harish and Dr. Rambabu, who have shared their life experiences. This is fantastic. I would like to thank Syngene leadership, Dr. Kenneth Barr, Head of Research Services, Dr. Jayashree Iyer, who is our Chief Scientific Officer, and a fantastic effort by Syngene commercial and branding team, Vivek Shah, Revti, Likesh Sharma, and many more. It's indeed a fantastic effort. I've been trying to share Syngene's vision on sharing the scientific learnings. Thank you so much for staying so long with us. We appreciate, and please remember, we will come back with our next episode of DMPK Unravelled seminar. Please look for the advertisements on our social media. Look forward to see you again. Thank you so much for joining.
Thank you. Thanks, everyone.
Thank you.